Thank you very much, Josh, for the introduction and also for the organizing committee of this wonderful meeting for inviting me here. I will just say it's always a privilege to attend this meeting. I always learn a lot that is not in textbooks from talking to families and seeing children and learning really what's important. So I've been given a monumental task this morning to talk about two very broad areas. One is what we know about or what's emerging in our understanding of the disease pathophysiology, simply what's the origins of this disease at cellular and molecular levels. And then pivoting from that is what are we doing and what is the progress in finding genetic therapeutics, things that might ultimately modify the disease course. So let's start with ATP1A3. What is it? It's the name of the gene that is associated with AHC and a variety of other neurodevelopmental disorders. It's a gene, it encodes, it writes the instructions for each cell to make a protein that is an ion pump. It pumps sodium and potassium ions. And this is a critical protein. Without it, cells can't generate electricity, like in the nervous system or in your heart. ATP1A3 is the one that's specific to generating biological activity and electrical activity in neurons that power the brain and our nervous system. And the way this works is it pumps sodium out of the cells in exchange for potassium. If you want to click to the next slide, I have a lot of animation, so this may get tedious, but yeah, next slide. It works now. Okay. Thank you very much. So a couple of key things happen. When it's moving sodium out of the cell and moving potassium into the cell, it sets up these concentration differences between inside and outside of the cell. Those are really important. Without those concentration differences or gradients for sodium and potassium, cells don't fire electrical impulses, which we call action potentials. So those are key things. The other important thing, and this comes up later in my presentation, is the number of sodium ions that are removed from the cell and the number of potassium ions that come back in is different. So there's this unbalance, if you will, of the charge that's moved across the cell. And that's important because we can measure that. That difference gives rise to a very, very tiny electrical current that we call the pump current, and that will come up. So what have I done that has destroyed this? So it works sometimes, and then it doesn't work again. So you want to click the next slide? I think we missed one. At least the vibration stopped. Yeah, so for those of you who are watching from home, you missed the good vibrations here in California. We were all wondering if we were having an earthquake to commemorate the event, but it turns out it's just a laundry dishwasher. Oh, sorry, a clothes washer. That we're probably on spin cycle, and that's why it was. Yeah. Okay. Anyway. Okay. Very good. Thank you, Bill. Anyway, so our approach, by the way, has been to primarily develop a set of cellular reagents that can be used in the laboratory to study human neurons with those ATP1A3 mutations that cause AHC. This is a technology that's been around for quite a while now. It's referred to as induced pluripotent stem cell technology, or iPSC technology. This is a very clever approach to take a cell from a person. We use blood cells, one can use skin cells, and then you change them genetically so that they can now be poised to become any other type of cell in the body. So we can take a blood cell, make an iPS cell from it, and then turn that iPS cell into a neuron that has the genetic cause of AHC already in it. And this is a remarkable technology that's been very useful for us and many other groups in the world. Okay, I don't know, it just keeps failing on me. Do you want to? Yeah, not getting anywhere. You can just click them. I'll just tell you when to change the slide. You can advance. So among our collection of iPS cells, and many in this room may be, their children may be donors to our project. You can see this is the list. I think this is the majority of the mutations represented in our collection. We have multiple cell lines on all of the 3 most common recurrent mutations listed here at the top, and we have some others that are less common. And from those cell lines, we can make 2 different classes of neurons that are prevalent in the brain. One set of neurons are the so-called excitatory neurons that give positive activity in neural circuits, and the other are inhibitory neurons that tend to dampen the activity. For a variety of logistical reasons, we've mainly done most of our work with the excitatory neurons. We've done some work with the inhibitory neurons, but suffice it to say that I think all the information I'm going to tell you today is largely from excitatory neurons. Next slide. So one of the major things that we do is we determine the electrophysiological activity of those neurons to try to get at the root cause of what's broken in AHC. We use a technique called the patch clamp technique. If you can go back, my slide before this is the one I want to cue off of. One ahead, thank you. There's a variety of things we can measure. The picture on the left is simply showing a couple of neurons in a dish, highly magnified, and that large triangular-like thing coming in from the side is a small electrode that's placed on the surface of the neuron and allows us to measure tiny, tiny electrical currents that we can then interpret and understand. So there's a long list of jargon here. I'll walk you through a couple of experiments that we do that has given us information about things. So first of all, as I said a couple slides ago, this asymmetrical movement of sodium and potassium ions gives rise to a small current itself. We can measure that current. We call that the pump current. It's sensitive to the inhibitor known as Wabane, which is a blocker or inhibitor of the ATP1A3 pump. And we can measure the pump current in the absence and in presence of Wabane to try to get at the sensitive component. That's simply our operational definition of the activity coming from ATP1A3. When we look at this in a neuron that does not have an ATP1A3 mutation, we get a certain level of Wabane sensitive current. Next slide. And when we do that in the in a neuron that contains a mutation, we see far less current. And here is just the quantification on the right. So one of our first observations was, and not unsurprisingly, that mutations in ATP1A3 associated with AHC impair the ability of this protein to pump sodium and potassium ions. Now that has consequences, and we've tracked those consequences now quite a bit. One of those consequences is these concentration gradients that are established by the pumping by ATP1A3 are not as robust. So under normal physiological conditions, this is a cartoonish of drawing of a neuron and a cell membrane of the neuron, and this large blob in the middle is the ATP1A3 doing its job, pumping sodium out of the cell, and that keeps the concentration of sodium inside the cell low while it's pumping potassium into the cell and causes the concentration inside the cell of potassium to be high. So normally, this is the concentration differences. Sodium is very low inside of the cell and very high outside of cells, and it's the opposite for potassium. This is the normal situation, and this basically sets up the electrical driving forces for firing electrical impulses in neurons that we call action potentials. Think of this as the battery that powers the nervous system. In AHC, we've made direct measurements of, or indirect measurements of what the internal potassium concentration is, and we were surprised to find that it was profoundly reduced. Instead of 140, it's 50. We didn't measure internal sodium. We're kind of inferring that that's probably higher than normal. And so what has happened already, just because the failure of the pump to move ions back across the membrane, these differences in ion concentration, which really power the nervous system, are blunted. Because of that, one of the first manifestations is the membrane voltage that the cell can maintain. We call that the membrane potential, is also blunted. Now here I have to divulge, I have to regress a little bit into some basic neurophysiology, which is all membranes have channels that allow either sodium or potassium to leak across the membrane. If the cell allows sodium to leak across, now this is separate from the pumping action of ATP183. If sodium is allowed to leak across, the membrane gets less polarized, or it's called depolarized. If, on the other hand, it allows potassium to leak out of the cell gets more polarized or hyperpolarized. And that's normal. And at rest, there's sort of a balance between these two effects. And it turns out that potassium is the stronger influence. And you find that most cell membranes, including in the nervous system, they're pretty hyperpolarized. But what happens if you change these ion gradients? Now I'm going to just change my cartoon there and show you that now instead of 140 of potassium inside the cell, it's only 50. That changes everything. That reduces the power that potassium has to get out of the cell, or the driving force, and that's going to limit its effect on polarizing the membrane. And so we've observed this now by directly measuring the membrane voltage with a little tiny voltmeter. And what we found was that in normal neurons, or neurons that do not have an ATP183 mutation, we can measure the membrane potential, and it's around minus 60 millivolts. Whereas many cells, but not all, of those AHC mutations, it's about 10 millivolts less polarized. Now, based on that, we thought, well, that's going to affect how the neuron can generate its activity, its action potential. And so this is simply a measure of the number of spikes that we see in neurons given a different level of stimulus. And so here we're just simply electrically stimulating the neurons and then counting how many times they fire. In this particular experiment, which is the mutation G947R, we see that in the normal neurons, or neurons without the mutation, fire like this, but those with the mutation have a reduced firing. And so this suggests that there may be an effect of this pump all the way through to altered firing of the neurons. And here's another example of that, where instead of just showing you a plot of the firing frequency, here is simply these are the actual firings in those cells, where little bit of electrical current. If you put no electrical current, you don't see any firing. And as you increase the amount of current, as we go down the slide, you see more and more firing until you get to a point where the neuron kind of fatigues. It fires so much that it's sort of, I don't want to fire anymore. And so you get these so-called action potential failures. That's normal. In these AHC neurons, again, that we've studied highly with G947R, we see failure much earlier at lower electrical input. And this makes sense. These neurons are less excitable electrically, and it's not unusual it was not unexpected to see that. And so what we've interpreted here is that neurons with low ATP1A3 activity have this greater susceptibility to failing prematurely in the firing. Another thing that we noticed, and again, we think we link this to this reduced concentration of potassium inside the cell, is the normal development of inhibitory neurotransmission. And this is the influence of those inhibitory neurons on other neurons in the brain. That is normally a developmentally regulated process. So early in development, immature neurons that receive a signal from an inhibitory neuron in the form of the neurotransmitter GABA are activated in a way that makes them more excitable. They will fire action potentials more readily. Well, that's not the job of GABA in the adult brain. In the adult brain, or the mature neuron, things have changed in such that when GABA is, the neurons are exposed to GABA, they are inhibited from firing. So this switch from GABA, the neurotransmitter GABA, from being excitatory to being an inhibitory neurotransmitter is linked to the amount of chloride ions in the cell. Now, where did chloride come from? Well, chloride, it turns out, the balance between inside and outside chloride is very tightly controlled in cells. And in an immature neuron, there's a lot of internal chloride in the cell. And so if you hit the cell with GABA, GABA opens up a pathway for chloride to escape. And as chloride is leaving the cell, the membrane potential gets less polarized, or so-called depolarized. And that's helps the cell fire an action potential. But as cells mature, there's an emergence of a new gene in those cells that transports potassium and chloride out of the cell. And that lowers the intracellular chloride concentration, such that now when you open GABA receptors, GABA doesn't leave the cell, it actually enters the cell because of this difference in the concentration difference between outside and inside. And in that setting, it's an inhibitory neurotransmitter. So this is often referred to as the GABA switch. During development of neurons, early on, GABA is an excitatory neurotransmitter, but as the neurons mature, GABA becomes an inhibitory neurotransmitter, which it will be throughout the life of the brain. So here's just the same diagram drawn a little differently. Again, immature neurons, they have high intracellular chloride and they're excitatory to GABA. But as the cells mature, they develop mechanisms to get rid of that chloride, pump it out, and now GABA is actually inhibitory with low internal chloride. Well, what happens in AHC? Well, we think there's a connection because normally we expect the potassium concentration to be very high inside of cells, and that helps push chloride out. But what happens in AHC when chloride is low? There's less push. So chloride is likely not to be pumped out as effectively. And so maybe intracellular chloride remains high longer in the maturation of neurons. And so we can actually measure that. Complicated slide, I apologize. But the bottom line is, when we look at iPS cell-derived neurons, and we look at what time point do they complete this GABA switch, here's a cell line without a mutation, and it's pretty much complete by about day 45. In neurons from iPS cells with G947R, it's delayed, and it never really maxes. It doesn't become maximally switched. And so we think this is evidence that the GABA switch is delayed in AHC neurons. And so just to put all this in a little bit of a context, again, for the neurons that we've studied, for the most part, we've seen failure of the sodium potassium ATPase ion pumping activity. That seems to be universal. That suggests that there's going to be changes in the ion concentration gradients in neurons. And we focused a lot on the role of depleted potassium in causing a depolarized membrane potential and a limited firing of neurons. And then I just mentioned that we see this delay in GABAergic neurotransmission to switch from being excitatory in immature neurons to being inhibitory in mature neurons. And sort of put this into context a little bit, I think the excitability of the neurons and the defect in that is probably tied to the attacks of hemiplegia and dystonia in some way. The connection is not 100% clear yet, but that's what we think. And then the delayed maturation of neurons in terms of the GABA effect may be related to developmental or neurodevelopmental delay. And that's not, would be not unusual for AHC. There are other conditions such as Fragile X syndrome and other conditions where the same delay in GABAergic switch has been shown. So this is probably going to fall right in line with that. Now, these are experiments done in cell culture. Okay, cultured neurons are not a brain. And so one has to wonder, will all this hold up if we were to look at an intact brain? So we have looked at an intact brain. In fact, we have made a mouse that has this same mutation, G947R. And this mouse is still under evaluation in our laboratory in collaboration with Dr. Jennifer Kearney's laboratory in my department. And one of the things we noticed early on is these animals were not healthy. They were smaller, both in weight and length, and they also didn't survive as long as their litter mates who did not have the mutation. And this is work that was all done on a particular genetic background strain known as C57 Black 6J. And that's a strain that other AHC mouse models have been used to create. And it seems to be a universal principle that mice on that background strain with ATP1A3 mutations are not healthy. Well, what else do these animals have? Well, first of all, we looked at pump current to make sure that the observations we made in tissue culture with human neurons was comparable. And so here is an experiment we did with neurons from the cerebellum, which are largely of the inhibitory neuron class. And we see, again, reduced pump current, substantially lower for the neurons we got from the mice with G947R versus their wild type or normal genetically litter mates. Now, what about the behavior of these animals? One thing was very evident was these animals are hyperactive. Now, anyone want to guess which animal is the one with the mutation? He's the busy one, running around looking for stuff. Very hyperactive. Now, looking in the cage, you get that impression, but we formally quantify this by using what's called an open field test, where we basically put them in an open box, plexiglass box with a camera, and we essentially let them run around as much as they want. And then we quantify how far did they run and how fast did they run. And so these animals run farther and faster. May explain why they're a little smaller, burning a lot more calories, but it's just part of their nature is they are hyperactive. The other thing, we put them through a battery of tests to evaluate their strength. And at baseline, these animals are weak. They have less ability to remove a sticker on their forehead, suggesting either a lack of strength or possibly coordination. If you suspend them by their tail, there's a procedure of quantifying their behavior that allows us to see whether they're weak or not. These animals have much higher scores, suggesting that their behavior suggests weakness. And then if we let the mice hang from their paws, their forepaws, from a wire mesh suspended above their cage, and then we time how long it takes them to fall, they fall much faster if they have the mutation. And that's, again, a quantifiable defect here. Probability of falling is higher, and they fall faster. So they can't hold on as long. So they have evidence of grip strength weakness. Another thing that was very obvious to us, again, by looking at them just observationally, is that many of the animals appear to have seizures. Now, there's a lot of behavior abnormalities these mice show. Some of it looks like maybe they have periods of dystonia, maybe they have periods of weakness. Those are two very challenging traits to quantify accurately. But they do have seizures. And so to really prove that, my colleagues in Dr. Kearney's lab inserted EEG electrodes into the mouse brain. And here's some things that they recorded. So there are periods of time when the mice are freely roaming around and appear to be moving normally. And this is pretty characteristic of that kind of resting EEG that we see in the mouse. On the other hand, during observed periods where they appear to have a seizure captured by video, we can go back and look at the same time when the EEG signal looked different. And here is an example of a seizure in one of these mice. And the video on the next slide shows a cage of mice. I think they're all ATP1A3 mutant mice. One of them right there is going to go through a behavior that's what we associate with a generalized seizure. This is different from the hyperactivity that I showed earlier. It's really wild running. And when it finally settles down, you'll see this brief moment where its hind legs just stick straight out. And that's a sign of a generalized seizure that is now kind of getting to the end. And so this was pretty evident, and not the only mouse. Many of these mice have really frequent seizures. So quantifiable phenotypes across the board. So that's the first part of my talk. And the second part is going to be pivoting to, well, what are we doing about this? And what can be done about it?
Join the meeting.
To frame this idea of genetic therapies or therapies in general, I took a step back and said, okay, well, how do you treat any disease, and in particular genetic disease? Well, certainly there's pharmacological approaches, and almost certainly all your children are on drugs of some kind to control the symptoms of the disease. Hold on one second. Okay. So, there's kind of two types of pharmacological treatment. If you wanted to develop a new treatment for AHC, you could look at developing a new drug that takes years, or you could potentially look for drugs that are already on the market, already approved, or at least have been trialed for safety in children, and maybe test those first because a lot of the work has been done. The challenge there is that's a limited number of compounds that you can test, and what are you looking for? It's just a challenge. So people are generally unhappy with pharmacological approaches in this condition. So the idea of doing genetic therapies, and there's a range of genetic therapies. This is just a small subset of things that are conceptually available these days for doing genetic therapies. One is we call gene therapy as general the term, but I like to think of that as gene delivery. Delivering a new copy, fresh copy, healthy copy of a gene into the organ that's affected by the disease. That's the traditional gene therapy. Another approach is using what are called antisense oligonucleotides. These are small synthetic pieces of DNA or DNA and RNA that can interfere with, hopefully selectively interfere with the expression of a mutated gene. And there's a lot of excitement in the neurodevelopmental disease world because of Spinraza, a drug approved for treating spinal muscular atrophy, has really transformed that condition. And there's many, many other active clinical trials right now with other antisense oligonucleotides. Then lastly, there's some newer approaches, gene editing or repairing the defect in the gene, and modulating the gene's expression are two others that are sort of slowly coming along. The major difference here is that pharmacological approaches treat the symptoms. They don't affect the primary cause of the disease, whereas the hope is that genetic or molecular strategies are disease modifying. They affect the primary defect, and maybe have a longer duration of effect. So when one has to think about genetic therapies, I think there's a series of very important questions that have to be answered. First of all, conceptually, will it work? Will it even work? Second of all, once you establish that it could potentially work, does it work? Can you prove that it actually does work in some system? All these are questions that have to be answered before you get to a patient. Importantly, is it safe? You don't want to create a therapy that's more dangerous than the disease. So treating these as very, very risky therapeutics, I think, is the right mindset. Lastly, if you get through all these and you get the FDA, convince the FDA to approve something, now the question is, who pays for it? Because a lot of these genetic therapies are expensive, particularly those involving viral vectors. And then if you can figure out who's going to pay, who can have it? Because that may also be some health equity issues there. So just, these are big questions. I'm not going to answer all of them. I'll tackle the first one and the second one a little bit. The way to approach, will it work? I think we have to know about the disease mechanism to be able to conceive of a strategy that something like a genetic therapy will actually work. And then does it work? Much more complicated, but there one has to figure out, how are you going to deliver this magical genetic therapy to the brain, which is one of the more challenging organs in the body to treat. You have to consider, how long will it last? Does it have to be dosed multiple times, or will once do it? And ultimately, does it work better than standard therapy? And so that's where clinical trials come into play. And to answer these questions about does it work, one needs key data from model systems, cellular model systems, animal model systems, ideally a little of each. So these are some things that, again, I think are actively being pursued in AHC. First one is the idea of, and there's not in any particular order here. Well, the order is actually determined by the duration of the therapeutic effect. But antisense oligonucleotides, the concept there is, can we turn off the mutated copy of the gene selectively so that just the normal copy exists in the cell? And that's something that, if that works, this would have to be delivered on a regular basis for the life of the individual, because the duration is short-lived. Maybe what we know from Spinraza, treatment of spinal muscular atrophy, the effect lasts for a few months, and then it dwindles. So one has to redose repetitively. And dosing these drugs, it's not given by a pill. It's not given even in a vein. It has to be given into the spinal canal. So that's a tricky thing to do. But it's become more and more routine, so it's not out of the question. Next on the list is gene delivery, delivering a gene, usually using some kind of viral derivative, a viral vector, if you will, to get it into the target tissue. In the case of AHC, there's a big effort to use the adeno-associated virus, also known as AAV. And that's a great virus for genetic therapies, because it's potentially has a duration of effect that's lifelong. Although there's a recent news in the muscular dystrophy world where the FDA just approved, I think yesterday, approval of an AAV therapy for that disease. There were some concerns about it, and the panel that was approving it was not unanimous in voting for its approval. In fact, there was, I think the vote was 6-4. Those that dissented said they weren't sure that it was going to be effective for the life of the child, because there's evidence that the effect dwindles over time. That may be specific to muscle, because muscle is turning over a little bit. Brain may be very different. So there's hope that treating brain diseases with AAV would truly be lifelong. So I would be encouraged about that. Lastly, gene editing, the ability to actually go in to the nucleus of a brain cell and actually fix the defect, is coming along. David Liu's lab in Boston has got an active program We're starting early in talks to collaborate with them to test their ideas of gene editing on ATP183 in human neurons. So I'm going to walk through a couple of these and just show you some of our own data. First of is the antisense oligonucleotide study. For those of you who don't know, these are small synthetic pieces of DNA or RNA and DNA combined that interfere with the ability of a cell to manufacture the protein encoded by RNA. And so this figure is illustrating what we call the central dogma of molecular biology, which is DNA contains the instruction set. It's converted to messenger RNA, and then messenger RNA is read by the cell to make protein. So DNA, RNA, protein. Where antisense oligonucleotides come in is you can short circuit this process, or you can actually abort this process by interfering with the ability of the RNA to survive in the cell. So messenger RNA is a single strand nucleic acid versus DNA, which is a double stranded or double helix. And so what antisense oligonucleotides do is they bind to the RNA in a targeted way and form a small double-stranded segment, which is not allowed in cells. And so this RNA DNA double helix is destroyed by an enzyme called RNase or ribonuclease H. And so this is one of the principles of using antisense oligonucleotides. You target a sequence for destruction. And in this case, it would prevent the synthesis of that protein in the cell. If you can make this selective for the mutation, which remember, in most cases differs by only one nucleotide, so it's not very different. If you can make this selective for the mutant allele, could this work? So just to show one more thing, there's now, again, what's being clinically trialed and approved by the FDA are antisense oligonucleotides that are designed a particular way. And that way is referred to as a gapmer. You may hear this term. It's a silly term. But what it means is there's a central core of DNA, usually pretty short, 10 nucleotides or less, that binds to the RNA and directs its destruction by ribonuclease H. But that's not very stable in cells. And so the clever design feature here is you flank that DNA core with some RNA bases, or actually not truly RNA, but chemically modified RNA bases, which protect the DNA core. And so that's the gapmer. The gap is the DNA, and it's flanked by these RNA bases. So that's the approach that we've taken. Now, conceptually, will this even work? Because if you knocked out one copy of ATP183 and you're left with only one functional copy, is that enough for the nervous system to work properly? So we tried to answer this question using the iPS cell neuron model. And what we did was we first started out by using this revolutionary technology, some of you may have heard, called CRISPR, which allows us to target a specific sequence in a gene for degradation. And so this is different from ribonuclease H. Here we're targeting the DNA in the cell. And what we did was we took one of our cell lines that had the common recurrent variant D801N, and we engineered CRISPR technology to destroy selectively the mutation carrying copy of that gene or mutant allele. And when we did that successfully, it took a while, we were left with a cell line that had only one copy of ATP183, and it was the normal copy or the healthy copy. So this would be the ideal situation if one were to use an antisense oligonucleotide approach, and you were able to selectively destroy the mutant allele. When we did that, we decided we need to measure a few things. We need to look at what is happening at the RNA level. Did we disrupt the balance in the cell in some way? And so we measured the RNA for ATP183. In this case, here is the level of RNA over time in culture from day 14 to day 35. which is the usual window when we do experiments. And this is the level of RNA in the original cell line that was heterozygous for D801N. So it's got two copies. One's the mutant copy, one's the healthy copy. Over time, there's not much change. But in this heterozygous knockout cell line that is missing the mutant allele, there's something else happening. It looks like the RNA levels slowly rise over time. We wondered about the protein. So we looked at the protein levels of the gene. And again, in the beginning, we saw evidence that there's less protein in the cell, which we predicted. We've knocked out half of the RNA, there should be half the protein in the cell. And that was the case. But over time, there is some adaptation or compensation by the neurons that allows the amount of protein in the cell to not be very much different when we get out to day 35 between the cell line that has two copies, one being a mutation, and the cell line that only has one copy. So this gave us hope that maybe neurons they're smart. They have figured out a way to compensate for the absence of one copy. So this bodes well if, in fact, we were able to make a perfectly selective antisense oligonucleotide that could only get rid of just the mutation. And that's been our goal. So we've been working on that for a little while. We've actually targeted the two most common ATP1A3 mutations, D801N. And here's an example of the kind of data that we use to screen antisense oligonucleotides. Here's an experiment with 20 or so antisense oligonucleotides that we designed and tested. Some of these seem to work pretty well. Slide, yeah. In other words, what we're seeing is we're able to knock down both copies. We're able to inhibit both copies, but slightly more of the mutant copy. So this told us two things. First of all, this gene is amenable to antisense oligonucleotides. It can be targeted. That's number one, and that's very important. Not all genes can be. Second of all, we can get some allele specificity. We can knock down preferentially the mutation copy versus the wild type copy, but it's not perfect. For D801N, we're probably around less than 3-to-1 in terms of the ratio of the two. Probably need to get, my guess is 9-to-1. So we need to keep working on that and design new designs to try to achieve that. We have also worked on targeting E815K with some seed funding from the 4 Henry Foundation. And here again, we have a range of potential effects, some of which look pretty good, with ratios probably on the order of 3 to 4-to-1 knockdown. So the gene is targetable. We can get some allele specificity. It's just not optimized yet. So more work needs to be put into this to get closer to that optimal 8-to-1, 9-to-1 ratio. So these are experiments where we're targeting the portion of the gene with the mutation in it. But there are other parts of the gene that could be targeted. So, as probably some of you know, genes are complex structures. This is now at the DNA level. They're made up of this sort of interrupted structure where there's some of parts of the gene which code for the protein. They're called exons, and other portions that kind of stitch the exons together are called introns. At some level, they're all made into RNA, but the cell then clips out the introns and just leaves the exons. But one could actually target anything in the gene with an antisense oligonucleotide and maybe achieve the same goal of knocking down a selective copy. And so with the help of Illumina, we acquired whole genome sequencing on 12 individuals with AHC, and these were 12 people for whom we had iPS cell lines, so it made sense. And this is a figure that simply represents the structure of the gene at the top, multiple copies of the structure. And below here are these lines that indicate the position of variants in the gene that are potential ASO targets. The red ones are ones in individuals that are Heterozygous, meaning there's only one copy of the variant. And so for us to be able to target something else in the gene, we have to know a couple of things. First, we have to know that in that individual, they have this genetic variant. And second of all, it can't be two copies of the variant, it can only be one copy. And we have to know, is that copy on the same chromosomal copy as the disease-causing mutation? That's called the phase. And that's tricky to figure out. Although with Illumina, we were able to figure out, in a couple slides, I'll revisit this, but we were able to figure out the phasing of some of these variants with the disease-causing mutation in some of these families. I'll come back to that in a moment. Now, again, with this funding from for Henry's Foundation, we were able to collaborate with the Charles River Laboratories, who reviewed this genome sequence for one individual and identify 65 potential targets for ASO development. Not all equal, some of them probably not very good targets, but we went down the pathway of saying, well, let's just try them all. So we've begun the process of testing oligonucleotides against all of those targets. And this is some data to date showing what we're seeing, comparing it to the ASOs that we started with, targeting the mutant allele. You kind of see there that that's what we call our benchmark value. And in this case, we're not measuring each allele separately. We're measuring simply the ratio, because that's sort of where we're going for. And what we're looking for are any ASOs that have a lower ratio than this benchmark. So far, we haven't seen very many. There's a couple here in the middle. Still working on this. There's another big batch coming that we hope to finish in the next month or so. But that's another approach, is if we can find something else somewhere else in the gene that's a common enough variant that might affect, that might be prevalent enough in the population that lots of people have it, then potentially that could be a solution to a large number of people with AHC, regardless of what mutation they have. There's a couple of drawbacks. One is we have seen some cellular toxicity from some of these ASOs, and that's one of the concerns about ASO technology, that these synthetic pieces of DNA have some toxicity to cells or to organisms. And so we were actually able to see that pretty readily in tissue culture. Here's a microscope field of neurons that have not been treated with ASO. And I know it really just looks like a mess, but if I show it higher magnification, it would look more like neurons. And here are two ASO treated batches of same neurons. Clearly there's something going on, and we think this is simply evidence of toxicity. One of the rules here is you want to test things as soon as you can, as fast as you can, but get to an answer of this is not going to work as soon as you can, because you want to reduce the number down to a more manageable number. So these are two that probably are too toxic to pursue. Now back again to the whole genome analysis and that we did with Illumina. Here we looked at the 12 or so whole genomes that we determined, and we looked at a few of the targets that we're already testing with ASOs, and we asked the question, if we were able to succeed making one of these targets into an ASO drug, what fraction of this small group of 12 would that be valuable for? And that simply stated, how common are these genetic variants in the population? Some of them are pretty common, and you can just see in this set, there's one here that's probably in 50% of all the cases that we had. Here's another one that's probably in about a third, and then some that are maybe much less frequent than that. But there's more to the story, which is, even though there may be that genetic variant in those people, the question is it in phase with the disease causing a mutation? And so that's this analysis. Here we show this is the actual nucleotide at that position. And number 7 here, the row labeled number 7, are the ones that we're targeting. And those are the nucleotides that our ASO will recognize. So even in this first one, where 50% of the cases have some variation at that position, only 3 of them share the targeted allele. Now, that doesn't mean we can't target the other allele, But if we were to proceed with a drug that targets this allele, 3 out of the 12 would benefit from that. So I've come to the conclusion, I think, especially for these common variants, D801N, E815K, and probably G947R, I think the money is best spent trying to get an allele-specific ASO on those mutant targets directly. That would have the largest potential benefit to the largest number of people. We're not going to abandon these. It's just I worry that there's a lot of effort for less democratization. Let's put it that way. The last thing I'll share with you is some preliminary data that some of you have already seen, and this is our effort to use the AAV vector that's been developed by the foundations to test in animal models, and we've been using it to test in our human neuron model. And this is just a summary slide that says, again, just a reminder that we're making iPS cells from individuals, from blood. Those get made into neurons, and these are then treated with the AAV. As a control, because we want to use a genetically matched cell line, we actually use the CRISPR system to change the mutant base back to the normal base to make a separate cell line. Now, this sounds like, wow, you've cured it in the dish. Yeah, but this is not the point of the slide. This is simply to make a reference cell line. Sure, proof of concept, but a lot has to go through to select very tiny populations of cells that have this correction without any other damage to the DNA. So we simply use it as a comparator. And here we are looking at the effect of the virus on the RNA levels in the cell. Can we boost the amount of healthy ATP1A3 messenger RNA? Can we boost the amount of healthy protein level for ATP1A3? And then if we can, is there a functional effect? So can we measure any effect electrophysiologically? So the first question is, can we boost the amount of healthy ATP1A3 sequence at the RNA level? And the answer is yes. There's a dose dependent. The more virus we put in, the higher it goes. If we just look at the ratio of the mutant allele to the wild type allele, we want to make that number go down. We want less mutation allele and more wild type. We can see, again, there's a dose dependent effect. The highest concentrations, we see a reduced ratio. We look at the protein level. The protein, unfortunately, we can't distinguish the protein from the mutant to the versus the wild type. We can only look at the total amount, but it looks like the total amount does go up with higher doses. So the next thing we did with an electrophysiological experiment, we took, I think, the middle dose range and then tested whether or not we could see differences in the pump current, which goes back to the first part of my lecture. Again, pump current is this basic readout of the ion pumping activity of ATP1A3. And again, in this case, we were funded by Hope for Annabelle. We studied the neurons that have this mutation L839P. At baseline, we can see differences in pump current. There was a significantly lower pump current in those cells. And then in the cells that we treated with the virus, which we had to select randomly because we have a way to tell which cells were infected and which ones were not. We still saw, despite all that, we still saw an increase in amount of pump current, nearly back to normal levels. So this suggests that we can introduce a copy of ATP1A3 using the AAV virus in cultured neurons and more or less restore normal pump current. Now, what's holding us back a little bit here is it's not a perfect experiment. We're kind of blind to what cells were infected, and that reduces the power of the experiment. And so we really would love a new vector that allows us to mark the infected cells so that when we're doing these single cell measurements like pump current or other electrophysiological readouts, we can pick the cells, that cell's been infected, that one hasn't, and then compare them in the same dish. So that's a need that we have at the moment. So just to summarize, I hope I haven't taken way too much time. In our hands, we see this common feature at the molecular level in neurons from either human neurons or mouse neurons, that there's a reduced pump current. That's the fundamental ion transporting readout that we have for ATP183. That impaired pumping leads to a disturbance in the ion concentration gradients in neurons. And those ion concentration gradients are critical for powering those neurons to be able to fire electrical impulses known as action potentials. We think that also impairs the switch from GABA, the neurotransmitter GABA, from being excitatory to being inhibitory, as in mature neurons. And that may relate to both the attacks of weakness and dystonia, as well as the developmental delay. Our mouse model, which I've never talked about in public before, by the way, you're a privileged audience to hear this for the first time. It's been slow. These mice have a clear phenotype. They may have a little more on the seizure side than other mice that have been reported, although I'm not so sure if those other mice have been interrogated carefully enough for seizures. But there are quantifiable features that those mice have, and that bodes well for understanding the disease, but also possibly for quantifying the effect of drug or gene type therapies. Lastly, we have this observation that we're repeating now. I want to repeat for different cell lines, different mutations, that neurons appear to be able to compensate when you remove the mutant allele by doing something to jack up the levels of the remaining healthy copy. And that really suggests that something like an antisense oligonucleotide approach has feasibility, and there's hope there. We know that using antisense oligonucleotides, we can target the gene and we can get partial allele selectivity, but that needs to be optimized further. And then lastly, we have shown, as I just showed you, that we can deliver AAV, a copy of ATP183 with the viral vector to neurons, and that has an electrophysiological effect. So anyway, this is the team. The major contributor to all of this work is Christine Simmons, who's in the audience. She takes care of these iPS cells. They're her babies. She does a lot of the primary molecular and genetic experiments, and none of this work would happen without her. She's assisted by Chris Thompson, who's the electrophysiologist on the team, John Mark Dick Kaiser, who helps a little bit with the molecular biology. And I mentioned Dr. Kearney. Her lab and her expertise is in genetic models of epilepsy, and her lab has basically adopted our G947R mouse, and I'm very happy that they've done that. I hope we can continue that collaboration with her and her colleague Nikki Hawkins. And then we initially started all this work with cell lines created by Kevin S. with help by resourcing some of the primary cell lines from patients from Kathy Swoboda. And I must thank all the foundations for their eager assistance with this work over the years, without which we would be nowhere. So anyway, thank you very much. Happy to take questions. Is there another microphone in the room? Okay, well, you.
Thank you, sir. I should be the one walking. So on one of your slides, you had a comparison of wild type to G947R pump current, and it looked like wild type was roughly 4x the pump current of G947R. I think it was the slide before the hyperactivity video. But do you have any guesses? I mean, one out of two genes are affected, but the pump current is 1/4. Do you have any guesses about that type of effect? What might be causing that?
So do you mean the kind of the quantitative effect? Yes. So yes, I get it. Yeah. So again, half of the genes are mutated. The pump current looks like it's reduced more than half in some cases, right? Yeah, I think this gets back to this idea that maybe the mutations have a dominant negative effect in some way. I don't think there's been a good molecular explanation for that. Maybe Kathy Swedner would offer one related to sort of her work on biosynthesis of the channel or the pump. So yeah, I think that suggests there may be a dominant negative effect. So I think that's the easy answer. So Would you agree with that, Kathy?
According to Hanna Poulsen's work, you really can't distinguish between a leak and a loss of current. So that dominant negative effect might just be a leak in addition to the current, the slippage.
Questions?
Two questions, I guess, is, with our disorder, a lot of it is alternating hemiplegia. So switching hemispheres of the brain, is there any of this data that contributes to one side, or basically the switching of the hemispheres? Is there like an abundance of positivity or a abundance of negativity that causes one side of the hemisphere or the other? In relation to that, assuming that it's neurological, and then being that sometimes it can contribute to upper body effect more than, the lower body. So is there any kind of research in relation to those causes and effects, as well as that being that potassium and so forth related, is that dietary as well, in relation to that?
Yeah, that's a really exceptional question. So I would just say, first of all, what I've shown you here, our experiments are very cellularly focused, and the brain is operating with lots of networks and circuits of neurons connecting to each other. Why is it right versus left or both? I think that's going to be a question that's going to take other measurements to figure out. And I think most of us have a sort of a gut feeling that it's probably some stochastic process, some random thing that happens that tends to predominate on one side or the other. I don't think we can learn anything from individual cells that would tell us about the sidedness of an attack. Your other question, very importantly, is, since I've focused a lot on potassium, don't run out and start feeding your kids bananas and apricots and stuff. I think our bodies have so many mechanisms to precisely control how much potassium is in our blood and in cells. This is really a phenomenon, a very local phenomenon, probably is irrelevant, except in the extremes. Certainly if someone decided they weren't going to have any potassium ever in their diet for the next 2 years, they'd be in trouble. The body can't compensate for that extreme, but subtle changes in the diet, probably irrelevant. So. Kathy. Well, it has to be picked up with the.
I'm going to take advantage. So Kenneth just brought up this interesting idea about the fact that high potassium levels extracellularly account for spreading depression. And the process in migraine marches much more slowly. And I think spreading depression is an electrophysiologic mechanism that does affect networks and could account for why this goes on and starts to affect different regions across days. And yeah, I mean, we talked to one family who, 3 days it affects the one side, and then there's a switch day, and then 3 days the other side. That's a common story we've heard over the years. So that mechanism. But the other thing I wanted to say is we just saw there's some interesting, I don't know if you're thinking about building like organoids, but there's a really interesting cerebellar organoid that's been created now, funded by the Pontocerebellar Hypoplasia Foundation, that is amazing. And when they express a mutation to make this cerebellar organoid, it's much smaller, like you get with pontocerebellar. It would be amazing if you could sort of replicate the mutation in a little bit more complex thing, because we know Purkinje cells are so affected, and be able to record from Purkinje. I mean, it's just an interesting thought, but I would love to see your AAV9 in your mouse model too.
Thank you, Kathy. Those are really great insights. So I think that spreading depression, or what the electrophysiologists like to call spreading depolarization, is almost certainly something that needs to be looked at in AHC in depth. And I think Arne van den Magnenberg in Leiden the Netherlands is really committed to doing that. And he's a world's expert on spreading depolarization. So he just needs to get his hands on the mice. He's working on that. So at the October ATP183 symposium that later this year in Chicago, Arne is going to give a whole session on spreading depolarization. So, but yes, I think you're right. I mean, experimentally, you trigger spreading depolarization by injecting a high local concentration of potassium or glutamate, I think. So yes, potentially some local disturbance in potassium clearance around neurons could be an issue, like it's thought to be in neurons. And actually ATP1A2, which is a cause of familial migraine, one of the thoughts is it's not able to clear potassium in the local environment. So your other question about organoids, we haven't done organoids. I hear about organoids all the time. That's a big commitment, and we're not ready to do that yet. There are people thinking about that, and Richard Smith, who joined my department last year, came from Boston. He has big interest in developing organoids, but mainly to look at cortical malformations. But he also has some ATP1A3 lines. So maybe at some point he can be persuaded to go that way. So anyway, I don't want to take up too much more time. Oh, we have one more question. Do we have time for that? Take it offline. Okay. All right. Thank you all very much. Thank you, people in the virtual world.